Hello, this is Mr. McLean, and I wanted to use this evening to take you through a few of the highlights of section 6.2 up through 6.5, which is the tour of the cell. So we're going to look at a few of the things that may be important to review when you're preparing for your test. Section 2, when we start off with this, it's going to just basically explain to us that there are two different general categories of cells. So you're going to have your prokaryotes and you're going to have your eukaryotes. And the main thing to keep in mind is that the prokaryotes are going to lack membrane-bound organelles. So they're going to be more of our our prehistoric type of cells, or the primitive cells. If you go back to the start of the life on Earth, they all started with prokaryotes. Eukaryotes are going to be much more complex, and that's really where most of our efforts are going to be spent in this particular chapter, just kind of looking at the different structures that are found inside of the eukaryotes. And of course, the biggest thing that you're going to notice whenever you do any type of comparison between these two different cells is that there are some similarities. So all cells are going to have a boundary, which is going to be your plasma membrane, also known as the cell membrane. They all have the cytosol, which would also be known as your cytoplasm. They're going to have some form of genetics, which are going to be the genes in different forms. They're not necessarily in the same form, but the coding is of the same format. And we'll have ribosomes. And one of the things to make a note of, whenever you talk about prokaryotes, they do not have membrane-bound organelles. If you look at a ribosome closely, a lot of times people consider it an organelle, but technically it is not. It does not have a membrane about it, so that allows it to be, consistent with the rules that prokaryotes do not have any membrane-bound organelles. If you look at the main features of a prokaryote, you're going to find out that they're basically not going to have any type of an obvious nucleus. You're going to see that there's not going to be any membrane-bound organelles. And, our easiest structure to kind of look at to visualize what a prokaryote is going to look like would be a bacterium. And if you look at this particular image, what we're going to notice is that the DNA is still there. In fact, it's more of a circular chromosome, but there's no boundaries, not a nuclear membrane like what we're going to see when we study the eukaryotes. Some of the other things that we had already mentioned, when you look inside in the cytoplasmic area, you will find ribosomes. You will find, in many cases, a cell wall, which is found in different types of eukaryotes, but the structures are going to be unique. From a eukaryotic standpoint, the big things that we're looking at are membrane-bound organelles, which we keep seeing over and over again, and they have DNA contained within a nucleus. So instead of those genes kind of just hovering in a particular nucleoid region, they're going to be bound very, significantly if we get into that. So just kind of following along, we have a whole chapter just dealing with the membrane, so I'm not going to get too much into that right now. But when you study the plasma membrane, biggest thing that we're going to notice is that it's a phospholipid bilayer. So each of these little guys right here, these are examples of phospholipids, and they're going to self-orient this way so that the portion, this gray portion, is polar and the yellow portion is non-polar. So what that does is you can figure that on the outside of your cell, you're going to have all this area, we're going to have water. And you're also going to have high concentrations of water inside the cell. So the orientation of the phospholipids is such that you have hydrophilic regions facing the water, and you got your hydrophobic region facing the internal area. We'll talk more about how certain molecules are able to pass through. We also have proteins that are embedded that go all the way across. A lot of times those are going to be our transmembrane proteins. And the other thing shown in this case right here with these little green molecules, those are carbohydrates, and a lot of times the carbohydrates are going to serve as a marker, so they're going to give us some type of molecular recognition capability. I think one of the most important things just to establish very early is that cells must be small to maximize the surface area to volume ratio. So if you look at this example right here, we examined this in class. You look at a small cuboidal structure right here, and just using this particular dimension of 1 by 1 by 1, that would give us a surface area of 6 and a volume that would be 1. And when we put that together, we get a 6-to-1 surface to volume ratio. And if we look at that same shape and we bump it up to a 5 and we do the math on all those dimensions, what happens is, yes, our surface area went up, but our volume went up dramatically larger. So as a result, this value went down. So if we're trying to maximize bringing materials into the cell, because everything you figure, if this is a real cell, all of our raw materials have to come in through the perimeter of that cell. So we want to have as good of a value here as we possibly can. So by taking that same cell in picture B, if you want to think about it, and bump over to C, we cut it up so that it has the same amount of surface area as we had here, and we're back to our 6-to-1 ratio. So that just addresses the question of why cells have to be so small. We want to maximize how efficiently we can move things both in and out of the cell. Now, it's going to be important. I would definitely reference page 100 as well as page 101. And we're going to see each of these structures as we go through our quick discussion, but those are pretty good images showing us the basic structures that are going to be found inside of a eukaryotic cell. And section 3 starts us off by looking at the nucleus. It talks about not only the nucleus, but also the ribosomes. So if you think about the nucleus, you're talking about the kind of the control center. The big thing about the nucleus is that's where the genes are going to be located at, and it's going to ultimately determine our heredity and how we make our proteins. Surrounding that nucleus, we're going to have our nuclear membrane. So if we zoom in just a little bit on this picture right here, what we're going to see is inside of here we have genetic material. This kind of coiled up collection of material inside of here is what we refer to as chromatin. Okay? Oh, doesn't quite stay where we want it to. But chromatin is DNA in a non-replicating form. So as long as this is during the interphase of the cell's life cycle, which we know it is because the nucleus is still intact, that's what you're going to see. Now, when you see chromatin kind of condense up and go into this kind of configuration, where you start to see, not a very good picture, but you start to see this is a chromosome, now you're talking about something that's going to be going through DNA replication. And we'll discuss that a lot more later on. Okay, a couple other things to point out. You're going to notice there's a darker region right there. That's our nucleolus, and that's going to produce ribosomal RNA, which is going to be used to make ribosomes. So we'll talk a little bit about why that's so important. The other thing is, take a look right here. I think this kind of shows it pretty well. But we have, the purple is where the cell or the nuclear membrane kind of ends, and this blue is where the ER starts at. Okay, so you can see that they're continuous. There's a connection point right there. And there's, of course, other continuous points throughout this. This is a double membrane. Okay, so it's going to be sort of like our cell membrane overall. And the other really important things are these pores right here. Okay, I must have somehow bumped that, so I'm not getting my my image modification again. But pull back down there. So our pores, if you look at it, they're going to allow those ribosomal components to be able to move out of the interior. You're also going to see RNA being able to move out of there. You're not going to see DNA moving through there. DNA is way too large of a molecule, so that's not going to happen. All right. So let me bump this down just a little bit. It's just talking a little bit about some of the internal components that we had just mentioned. All right. So taking a look at what the ribosome actually is, we're going to see that ribosomes can be found in so many different locations. You're going to find them in eukaryotes. You're going to find them in prokaryotes. You're going to find them even within certain organelles, such as the mitochondrion and within the chloroplast. And the big thing about the ribosome to keep in mind is it's made up of two different types of material. It's made up of that RNA, ribosomal RNA, and it's made up of protein. And that's where you're going to make your protein synthesis possible. So we have two different areas where you could potentially find ribosomes. You can find it in the cytosol, and the ones that are in the cytosol are classified as free ribosomes. And these ribosomes are no different than the ones that are bound. They can actually flip between the two locations. In fact, we're going to learn a little bit later that the genetic material is playing a role in determining where they go. But the idea is, if the ribosomes remain in the cytosol, the proteins that are manufactured there most likely will stay in the cytosol as well. The ones that are bound to the ER are probably going to make proteins that are destined for release, either as secretions or part of the plasma membrane. We also do have a situation where they can become lysosomes. Now, this picture shows us that a ribosome is actually two parts that come together. So if we were to take our messenger RNA and kind of put this in here like this, and we're going to see this a lot more like in 16 and 17, but messenger RNA is going to have codons of information, and there's actually going to be different components within this, different sites, as we would say. And what we're going to do is we're going to have transfer RNA that's going to come in here, and it's going to bring together a sequence of amino acids. So as we kind of build that primary level of structure, you can kind of see how the ribosome is going to be able to help and assist in that